Good morning, good afternoon, and good evening, everyone, from wherever you're joining us. And welcome to the 12th and final webinar in the Women's Health series. Today's webinar is titled Hypertension and Hypothalamic Changes in Menopause, featuring Dr. Teresa Milner, Professor of Neuroscience at Cornell. Today, Terry will share her research that uses a mouse model of accelerated ovarian failure to explore estrogen's role in menopausal hypertension. Before we get started, we'd just like to acknowledge our partners for this series, the American Physiological Society, the Society for Women's Health Research, and Scientist.com. We'd also like to say thank you and give a shout out to all of our series sponsors. None of this would be possible without them. And we'll share a special message from today's sponsor, Alzet. Alzet, now a subsidiary of Lafayette Instrument Company, has been providing Alzet pumps and accessories for almost 50 years. Over 22,000 publications demonstrate their ability to reliably deliver agents continuously to lab animals at precise rates, regardless of molecular weight. We would like to thank Dr. Teresa Milner for trusting our pumps to facilitate the important work she has done, including but not limited to what she's about to share with the audience. We hope that you enjoy the webinar. I'm Sarah McFarland from the events team here at Scientist.com, and I'm happy to be your host for today's event. Before we get going, I wanted to cover a few housekeeping notes to help you get the most out of your experience today. First, take a look at that interactivity panel at the right of your screen with 4 tabs: chat, Q&A, polls, and handouts. This is where you'll participate in the Q&A as well as access all webinar resources. We want this to be an interactive experience for you, so please share your questions, thoughts, and comments using that Q&A tab, and we'll try to address as many as we can at the end of the webinar today. You can also check out that handouts tab for some links and documents to help you learn a little bit more. And lastly, if you're having any technical issues, please try refreshing your browser and ensure that you're using Google Chrome to access this event, as it is the most stable browser. If you're still having trouble after you refresh, use that private chat function to communicate with me by sending a chat to Sarah, and I can help you to troubleshoot. Okay, and without any further delay, I'm going to bring Terry on here. Hi, Terry. Thanks so much for being here with us today, and take it away whenever you're ready.
Great. Thank you so much, and thank you for the kind introduction. I'm happy to speak to you today on my work on hypertension susceptibility and changes in hypothalamic plasticity in a mouse model of menopause. So ovarian hormones, particularly estrogens, can impact brain functions at all ages. And I know you've been hearing a lot about this through the series, but they can affect learning and memory, affective state and mood, motor coordination, responsiveness to stress, and blood pressure regulation, as well as pain. And today I'm going to speak about effects on blood pressure regulations. So there are known gender differences in the pathophysiology of hypertension. First of all, young men have increased rates of hypertension, that's high blood pressure, compared to age-matched women. And the incidence of hypertension increases in women compared to men after the age of 45 as they approach menopause. And we do know that central mechanisms in the brain are critically involved in these processes. So a brief review on perimenopause and hypertension. We know that perimenopausal period, which is the ages between 45 and 54, this is when estrogen levels are beginning to fluctuate here, as shown here. And it's also a time when there's an important onset for menopause hypertension susceptibility. And we do know also that the responses to antihypertensive drugs can differ between the genders. So in our mouse models, we've been able to recapitulate these sex differences in hypertension in young mice. In our earlier studies, we showed that if you give osmotic mini pumps containing angiotensin to young male mice and young female mice, the males, as shown here, have increased blood pressure, where the females do not. And other labs have also recapitulated these results. Studying perimenopause in rodents can be difficult. So typically, human menopause, as shown here in the schematic, the estrogen levels fluctuate. They fluctuate when they hit perimenopause, and then in postmenopause, they are reduced to negligible levels. And at this point, too, in postmenopause, women stop having a menstrual cycle. So typically the models that have been used to study menopause in rodents have either used ovarectomy, which removing the ovaries mirrors human surgical menopause, but it's abrupt and it lacks the hormonal changes that are seen in the natural progression to human menopause. Another model that's typically been used is natural aging in rodents. And rodents do experience an acyclicity in their estrous cycle, but they don't have a total loss of estrogen. And this is likened more to an estropause, where the estrogen levels are more in a mid-level here compared to humans, rather than the negligible levels seen here in menopause. So for our studies to get at what's happening in both the peri and postmenopausal periods, we use what's called an accelerated ovarian mouse model of menopause. And this uses a chemical called 4-vinylchlorohexene dipoxide, or VCD for short. And VCD was discovered as a byproduct of rubber tires. Women who worked in rubber tire factories or lived next to rubber tire dumps went into premature menopause. And it turns out VCD selectively kills the primary follicles of the ovaries. There have been many reviews about this process, but it was originally discovered by Hoyer and Mayer. And what they found is that if you gave very, very low doses, 160 milligrams or even a little bit lower of VCD for short periods of time to young female mice when they're starting to mature, around 2 months old, you can actually mimic what both peri and postmenopause. And what happens is VCD selectively targets the primary follicles in the ovary without affecting the peripheral tissues. And this results in a natural apoptotic process of ovarian follicle atresia, as shown here in this movie. So as VCD is given, it comes in and starts killing the primary follicle pool. The primordial follicle pool then replenishes it, but eventually you run out of the primordial follicle pool, and subsequently the primary follicle pool. And with the removal of these the follicles and the lack of mature follicles being generated, you can actually time out periods where the hormonal changes resemble what you see in peri and postmenopause, as shown here in this cartoon. So the AOF model now can be used to study perimenopause. So if you give VCD here at this period, eventually you start getting estrogen levels kind of fluctuating, but also the estrous cycle is irregular, and you have some high or much lower levels of estrogen than you normally do have. And then by the time you get time out to what's called post AOF, there's the estrous cycle has stopped and the estrogen levels are negligible. Similar to what you see in human menopause here, shown in this cartoon, where the perimenopause women have fluctuating levels of estrogens, as well as an estrous cycle being irregular, and then a stopping of estrus of the menstrual cycle, as well as a loss of estrogen. But the nice thing about this model, the VCD model here, is that it's not abrupt, and it does model what is happening to the other hormones, particularly LH, RH, progestin, androgens, that is similar to what happened in human menopause. So using this model, we then asked the question in our AOF mice at a perimenopausal time point, susceptible to this slow pressor angiotensin induced hypertension. So just a little bit about the experimental design of these studies. Mice, female mice, we had 6 to 8 per group. They were either given oil or VCD injections for a period of 3 weeks, 5 days on, with the rest period of the weekends. And after that time, we then either at 50 days post VCD injection, which is correspond to the peri-AOF period, we implanted the mini pumps with the angiotensin or controls for saline, and then euthanized them 2 weeks later. And this is similar to what we did in our previous studies with young males and young females. We also studied post-AOF mice, and at this point, it was 139 days when the mice are approximately 6 months old, where we implant the mini pumps, and then 2 weeks later, collect their brains. So for this, the exciting thing we found was that both peri and post menopausal or AOF mice had increases in blood pressure with angiotensin, similar to what we saw before in males, but the controls with either oil or VCD at a time point where they still have primary follicles in their ovaries, they did not have an increase in blood pressure. So then we are interested in asking, how do these blood pressure elevations in AOF mice affect the brain? So just backing up a little, we just wanted to discuss the ovarian steroid receptors. They're all over the body, and not only in the brain, but also in the ovaries and uterus. And we do know that what happens in the brain through its hypothalamus and its descending projections to the pituitary influence hormones that influence the output of the ovaries, but then the ovaries feed back into the brain to affect these as well. Estrogen receptors are known to be in 3 major categories: estrogen receptor alpha, estrogen receptor beta, and GPR1 or GPR30, as it's also known as. Progestins, progestin receptor A and B, and androgens, androgen receptor. Ovarian steroid receptors in the central autonomic pathways are particularly prominent in all parts of the pathways, including the paraventricular nucleus here, as well as the nucleus of the solitary tract, the rostroventral medulla, and the PVN, which receives inputs from the subfornical organ, then projects directly out to the intermediolateral column of the spinal cord, where it then can affect the baroreceptor reflex. Within all these brain regions, there's different types of signaling of the ovarian steroid receptors. There's what's called genomic signaling, where the steroids bind to nuclear receptors to influence gene transcription, as well as non-genomic signaling, where the steroids bind to extranuclear receptors to activate cell signaling pathways. And both types of signaling are important in regulating hypertension. An example of what in the hypothalamic paraventricular nucleus, or PVN, the prominent receptor there is estrogen receptor beta, and shown here in this fluorescence micrographs, estrogen receptor beta from an EGFP mouse co-localizing with fluoro gold retrogradely transported from the spinal cord, showing that there's high overlap of ER beta neurons with neurons that project to the spinal cord from the PVN. We do know that this projection modulates sympathetic and neuroendocrine systems, which are pivotal for the blood pressure regulation, and that, as I mentioned, that they project down to their spinal cord. We did have a review paper showing that there are sex differences in both the distribution of gonadal steroid receptors relative to ER beta containing neurons in the PVN. And this is just a summary diagram from this. What I want to draw your attention to is if we look at nuclear receptors, depending on whether we're looking at neuroendocrine subregions or autonomic subregions, there are slightly different distributions in these different receptors, so shown here in the color coding, depending on whether you're looking in male or female mice. In particular, the estrogen receptor beta is much more prominent in the neuroendocrine regions than in the females and the males, and that in autonomic subregions, they're about the same. However, nuclear androgen receptors are co-localized with estrogen receptor beta in the male mice. At the electron microscope level, we have shown that ER beta containing neurons, shown here in the purple, receive inputs from androgenergic receptors, as well as having the nuclear receptors, but that extranuclear receptors can from both androgen receptors and GPR1 are on the membranes of these ER beta neurons, as well as androgen receptors are also contained in glia. And in the females, they receive more inputs from both estrogen receptor alpha and progesterone receptor containing afferents. Previous work in males have shown that in the PVN, the glutamatergic NMDA receptors are crucial for mediating hypertension. Upregulation of NMDA receptors in the PVN spinal neurons enhances excitatory drive in many hypertensive models, as well as activation of these NMDA receptors in the PVN can stimulate NADPH oxidase activity, which generates reactive oxygen species and activates L-type calcium currents. In males, PVN NMDA receptors are, as I mentioned, crucial for hypertension. In another study performed by Dr. Glass, who is also my principal investigator on my current grant studies on the that I'm presenting here, but he did a study where he showed that if you did spatial temporal deletion of NMDA receptors in the PVN, this actually attenuates slow pressure angiotensin and dampens local ROS production. We also have done many studies in the PVN on different subtypes of neurons that have been shown to be involved in blood pressure regulation in terms of what's happening with the GLUN1 subunit of the NMDA receptor. And as I mentioned, ER beta we know is critical. In young males, if you give, it increases blood pressure with the angiotensin, and young females do not. What happens with ER beta is they actually kind of reflect the GLUN1 subunit of distributes differently in males and females. When in the hypertensive males, they actually increase on the membrane, and on the young females, they do not. Surprisingly, angiotensin 1A receptor, corticotropin releasing factor 1 receptor, and arginine vasopressin subunit P47 FOX, none of these had a change that reflected the blood pressure change. And this became important for our further studies because we ended up just focusing on estrogen receptor beta, since there was a critical relationship between blood pressure regulation and the trafficking of GLUN1 within these cells. So then our next question was to ask, are the hypertensive responses of these AOF mice at peri and postmenopausal time points reflected by the trafficking of the NMDA receptors, particularly the GLUN1 subunit, in estrogen receptor beta containing PBN neurons? So for this, we primarily used a anatomical method called dual label immunoelectron microscopy. And as illustrated in the schematic on the left, we take an antibody to ER beta, and it is identified using a diamino benzidine peroxide reaction product for the ABC, which is a more granular, which I'll show you in the next example in the next slide. We then combine this with GLUN1 labeling using silver intensified immunogold. And what's nice about the silver intensified immunogold, it gives us very discrete reaction product, which then can be quantified, so we can tell where this receptor is localized within a cell. And this schematic on the right shows what a subsection of a dendrite would look like. And if you found immunogold reaction product for GLUN1 on the membrane, this would be available for ligand binding. In the case of GLUN1, it would be glutamate. Also near the plasma membrane receptor, which would be in this compartment, it would be ready to be inserted into the membrane. And the cytoplasmic receptor localization would be more responsible for degradation, recycling, rather than ligand binding. So when we did our studies, we looked at the PVN ER beta neurons where they contain GLUN1. Shown here on the left is a coronal section through the paraventricular nucleus. In our we have these ER beta EGFP reporter mice. This is the reaction product for ER beta. You can see there's many neurons, especially throughout the PVN, but especially prominent in the autonomic regions. And in this example of an electron micrograph, this is a dendrite labeled with ER beta EGFP reaction product, and you can see it's sort of granular and fills the whole dendrite. And these discrete black dots are silver intensified immunogold. The one on the membranes here are shown here, this example. Near the membrane is this example, and in the cytoplasm, here's two examples here. So again, just quick experimental design for these studies. We had 8 animals per group. The VCV injected mice were injected at a peri-AOF time point around 56 to 58 days, with either implanted with mini pumps containing saline or angiotensin. Then we had VCG injected mice from post-AOF time point, either with mini pumps of saline or angiotensin, and then age-matched oil-injected controls from both the peri and AOF time points. Data collection and analysis was conducted by a person blind to experimental condition. This is really important for not introducing bias to your analysis. And we randomly selected 50 dendrites that were dual labeled for GLUN1 and ER beta GFP per mice that were photographed on an electron microscope and analyzed with MSID software, which is kind of a high-end ImageJ software for giving us diameter and area. And then we did two-way ANOVA followed by Tukey post-hocs to compare the GLUN1 sigma particle density. So the main result from this study was that slow pressure angiotensin differently affected the distribution of GLUN1 in the ER beta EGFP dendrites depending on the AOF time point. So if we looked at the mice, AOF mice, here's an example of what the micrographs would look like in terms of the reaction product for GFP as well as the GLUN1 silver intensified gold. But if we looked at the compartment of plasma membrane, both near and on the total, the peri-AOF mice had an increase in the number of these receptors on the membrane, where the post had little change. The important thing about this is the more receptors on the membrane, the more they would be available for glutamate binding. If we looked at the cytoplasmic total, there was actually an increase in the oil mice and a decrease in the post dendrites from the post mice. We then looked at ROS production, if it's increased in these ER beta, GFP, PVN neurons in the post mice following slow pressure angiotensin. And we did this because in males, NADPH oxidase generated ROS has a critical role in angiotensin-induced hypertension and is associated with NMDA receptor activity. And we also then know that ROS production in these neurons is measured both pre and post mice following saline or slow pressure angiotensin infusion. And these studies were showed that ROS production is increased in isolated EGFP, ER beta containing neurons in the post mice. And here's some examples of what they would look like under the microscope. This is what happens when we either gave vehicle or NMDA, and these studies then showed a increase in NMDA in the ROS production following angiotensin. So as a summary of these studies in this schematic, we have looked at both non-AOF female mice, peri-AOF, and post-AOF mice. There are some baseline differences as shown here in this schematic, but also when we add angiotensin, the important thing is that in the peri-AOF mice, there's an increase in the membrane-associated receptors, and this corresponds with both the increase in blood pressure and the fluctuations in the estrous cycle that are irregular. And in the post-AOF mice, not shown was that I didn't show was that there was actually a kind of a ballooning of the dendrite size, suggesting that receptors as they get inserted are not being trafficked out and the membrane is growing. But there's an increased ROS production, but not much of a change in terms of the GLUN1 receptors associated with the membrane. So then the next question we asked is, we now have looked at GLUN1, but we wanted to know if the AMPA receptor subunit, GLUA1, trafficking was altered in these neurons following slow pressure hypertension in the peri and post AOF mice. And the region we focused on, this receptor subunit, it's recruited by NMDA receptors to regulate hypertension in the PVN. And we did this in the same tissue that we had collected from the in our previous study using the same experimental design. What was striking to us when we first looked at this is that really the major changes were in the post AOF mice, as you show here in these results. If we looked at just the saline treated animals, there was not much of a difference in distribution of the GLU-A1 subunit until you start looking in the post AOF mice. The young females, the males, and the peri-AOF mice were similar. We then looked at the plasma-lemma GLU-A1 after angiotensin and found that the biggest changes were in the post-AOF mice after angiotensin, where we now have an increase in GLU-A1 particle density. And I just wanted to mention this has become a focus of our current studies, which I'm not going to talk about much today, but it does point to a different receptor being involved in the mechanism that prevents hypertension in both the peri compared to the post AOF mice. We also found in these mice there is a change in cytoplasmic GLU-A1, but mostly in the non-AOF controls, where they had an increase. So as a summary from these studies, the distribution of GLU-A1 is similar in males and non-AOF females. With the peri-AOF mice and the post-AOF mice, they start out very similar as well. However, when you give angiotensin, it's these post-AOF mice that have more of an increase in the GLU-A1 subunit associated with the membrane. At the same time, when these mice are hypertensive as well as acyclic. So just as a summary of where we are so far, with that part of the story was that GLU-N1 trafficking and GLU-A1 trafficking within these ER-beta EGFP neurons in the PVN differ depending on the AOF stage and as well as the sex. So in males, GLU-N1 increases on the plasma membrane. There isn't much of a change in GLU-A1. They do have an increase in blood pressure. Young females, not unexpectedly, they have a decrease in the plasma membrane GLU-N1, an increase in cytoplasmic, but there's no change in blood pressure. Both peri and post-AOF mice have increase in blood pressure. However, GLU-N1 increases on the plasma membrane of the peri-AOF mice, where GLU-A1 increases on the plasma membrane of the post-AOF mice. So the next steps, we've identified peri-AOF as a critical period when both hypertension and adaptations in glutamate plasticity in ER-beta membranes occur. So then the next phase of our grants, which was the second renewal, we looked at whether ER-beta influences the susceptibility of hypertension and NMDA receptor plasticity on select PVN neurons in the peri-AOF mice following slow pressure angiotensin. So for this, we wanted to know if cyclic administration of ER-beta agonists in these peri-AOF mice affect hypertension due to slow pressure angiotensin. And we chose the menopausal time point, because we know that's when GLUN1 is critically involved in the mechanism promoting hypertension. And what do I mean by cyclic? At the time we gave the pump implantation shown here in the schematic, we then gave hormones, but every 2 or 3 days instead of every day. And we gave low doses, so it wasn't like they weren't very high. They were more physiological. And we measured blood pressure using tail cuff, mainly because we wanted to keep track of these during our when we fix the brains. We want to make sure that the brains aren't compromised by anything we've done surgically. And then at the end of this 2-week period, the animals have been given not only angiotensin mini pumps, but the hormones and blood pressure is monitored when we euthanize them. So the experimental design is similar to what I showed in the previous study. What we found was, it was very exciting. The cyclic ER beta agonists attenuated blood pressure increases in the peri-AOF females, but not the males. And shown here in this example, when we gave saline in the saline vehicles, or we gave vehicle estrogen instead of ER beta agonists, we had a vehicle. They got hypertension in both the males and the females, and we gave 3 different agonists. This result shows that for DPN. We also did ER beta 041 and ligurgiginin, which are 2 other agonists which yield similar results. With DPN, you can see here, with the angiotensin, the blood pressure is brought down to normal. However, it did not affect the males. So then we characterized changes in PVN EGFP neurons following this agonist replacement using methods similar to what we had done before. In this case, we looked at the GLUN1 redistribution using immuno-EM, NMDA-induced currents, ROS production, as well as in this study, we did spatial temporal deletion of estrogen receptor beta in the PVN. And just another picture here showing the distribution of ER beta neurons in the PVN. So consistent with a decrease in blood pressure to normal levels in the female mice, the BCD treated mice had did not have an increase of the plasma membrane associated GLUN1s, where the males did. And the VCD treated angiotensin mice with the DPN had less neoplasmal GLUN1, as well as less cytoplasmic and less total, where the males had no change with the DPN and angiotensin. And this is just example electron micrographs shown over here on the left, showing the distribution of these immunogold particles in a vehicle and a control DPN treated animal. We then found that DPN also reduced the NMDA-induced currents in the ER beta PVN neurons in angiotensin peri-AOF. And these experiments were performed by Gong Wang, our collaborator. And what he showed here was in the vehicle mice, you have your NMDA-induced currents, but if you have the DPN plus angiotensin, the induced currents were much reduced. We also looked at ROS production in these mice and found that DPN inhibited the increase in ROS production in the ER beta GFP PVN neurons from angiotensin peri-AOF mice. Shown here is example, especially over here on the right, where we had females with labeled with the ER beta GFP. Here's DHE, which is the measure of the ROS, and we had a decrease in this ROS production in the angiotensin plus DPN animals compared to the angiotensin vehicle animals. We then did spatial temporal deletion of ER beta and the PVN. And for these, we actually started out with non-VCD animals. We just wanted to see if the presence or absence of ER beta affected resting blood pressure. And we started with a unilateral injection just to see, because we do know that these neurons are critical for blood pressure regulation, and also sometimes when you knock out both, the animals may not do so well. So we just started out with a unilateral injection. And we were surprised, but also happy to see that just removing ER beta on one side of the PVN using an AAV virus, Cree virus injected into flox mice, was that the control blood pressure changed in the female mice in particular. So here's the post-injection, and it wasn't a huge increase, but it wasn't a significant increase. And with the mice, where the males actually went the opposite way. So then we continued in the next phase. We just looked at the unilateral deletion of ER beta PVN in the PVN, and how it affected blood pressure in mice suffused with angiotensin. And shown here that in the young mice, which normally don't get hypertensive when they're given angiotensin, in the absence of ER beta, they actually did get hypertensive. We then since repeated these using bilateral injection, and it was similar, and we didn't show that in the paper, but the results were similar to the unilateral. Just to show the histological follow-up, here's the injection of the labeled AAV virus unilaterally in the PVN. We then quantified ER beta neurons using immunocytochemistry and found that here's the uninjected side versus the injected side on both the males and the females. And you can clearly see here there's a loss of cells. And when we quantified them, the cell density of ER beta neurons was decreased, and similarly in both the males and the females. We then used a Nissl stain to also look to see if it was just an effect on loss of all cells, or it's just the ER beta expression, and there was no difference in the number of Nissl labeled cells in either the males and females after the Cree injections. So as a summary and conclusions from these studies, we took our perimenopausal mouse model, the peri-AOF. When we gave them angiotensin, blood pressure was increased, and if we gave vehicle, it was still unaffected and still increased. In the schematic of an ER beta containing dendrite, we had an increase in NMDA receptor on the membrane, as well as an increase in NMDA-mediated currents and an increase in ROS production. if we had these peri-AOF mice with angiotensin, but then gave cyclic administrations of ER beta agonists, blood pressure was reduced to normal. And if we looked at the PVN of the dendrite, the NMDA receptors on the membrane were much reduced, more to normal levels. NMDA-mediated currents were reduced to those of controls, as well as NMDA-mediated ROS production was also reduced. So these studies then demonstrated that dysregulation of estrogen contributes to an increase in the sensitivity of these peri-AOF mice to angiotensin-induced hypertension, in part by modulating a signaling pathway involved in ER beta and NMDA receptor in the PVN neurons. So currently, we're working on our most recently renewed grant, and these studies are all in progress. But we're looking at mechanisms that contribute to the post-AOF hypertension. As I mentioned in our earlier study, we found that AMPA receptor, the GLU-A1, was prominent in the, which is prominent in the PVN, is important for the mechanism promoting hypertension in the post-AOF mice. We then had preliminary data, which we've now confirmed that GPR receptor is more important than estrogen receptor beta in promoting post-AOF hypertension. In this schematic here, these photographs here are RNA scope of the GLU-A1 and GPR, showing the overlap with another population of neurons containing oxytocin, which we know from the literature is oxytocin is also contained in ER beta. So ER beta and GPR are in the same cells, but they're shown here in the merged diagram, but they are regulating more of the post-AOF hypertensive susceptibility. So that story is to be continued. And with that, I just want to many thanks to my collaborators and the many students who have involved in this project. In particular, Dr. Michael Glass, who's been the co-PI on the grants with me to study the hypertension effect, and then many students here, Natalina Contarughi, Costantino Idacola, who's the head of the Brain and Mind Research Institute here, my postdoc, Jose Marquez Lopez, and my student, Tracy Van Kempen, as well as Sonora Mazid, and Gong Wang, who did the physiology study, Claire Woods, who did a lot of the ROS studies, and Fang Mingyu, who helped with a lot of the quantification. I also want to thank my long-term collaborator, the late Bruce McEwen, who helped us design the hormone replacement studies, as well as Elizabeth Waters, who helped a lot with these studies looking at estrogen receptors in distributions in the PVN. And then we've had many students, both at Cornell University, Hunter College, who have helped with the various quantification of the electron microscope studies. And our collaborator, Ken Karash, formerly at the National Institutes of Health, who provided the fluxed mice for ER beta, as well as helped us design the hormone replacement studies at the last few years. And my grant support from Heart and Lung, as well as National Institute of Drug Abuse, the Neuroanatomy M-Core here at Cornell, which I direct, but also has the electron microscope. And we also want to acknowledge the summer programs, especially the Weill Cornell Summer Access Program and the Gateways to the Laboratory programs, where a lot of our students have come from over the years. Thank you.
Thank you so much for that, Terry. Such a great presentation. I'm going to let you take a moment to take a sip of water and catch your breath while I just have a little chat with the audience.
Great.
So I just want to remind the audience that the Q&A tab at the right of your screen is where you can submit questions for Terry. And if you do have to leave early, I do have a little pop-out link to our survey here. We'd love to get your feedback on your experience today, and we do read all of your responses. So thank you for attending live. Okay, and so let me grab Terry back in here. Hi, Terry. Thanks so much again for that presentation. And if you're ready, we're going to jump right into the Q&A.
Great, thank you.
Okay, first question that we have here is actually from a past webinar speaker in the series from Paris. So she also uses the AOF model to look at impacts on skeletal muscles. She's wondering if your mice at peri-AOF and post-AOF are age matched.
So the one slide I showed, I can go into more detail on that. So they all started out the same, and then we had some, they were actually injected at the same time, the same age. And then the mice that were peri-AOF were given implants around 58 days. And the mice that were post-AOF were given implants around 139 days. And at the same time, we had, in addition to VCD treated, they were given, we had another group that was given oil and euthanized at exactly the same times, as well as the males. The males also were carried along with them. And many of these were actually litter mates too. So we split them up that way as well.
Okay. Perfect. I think you might have touched on this at the beginning of your presentation, but someone has asked, are there other models of menopause that you use, even if they're not in mice specifically? And what are the relative benefits or features of using those versus the AOF?
Well, right now, in terms of mimicking peri-AOF and post-AOF, the VCD model is the only one that actually does that. But I have used aging mice, and I've had used overectomized mice in earlier studies.
Okay.
As well as natural cycling young mice. we look and see if, you know, if the if hypertension varies depending on estrous cycle stage. So,
awesome, thank you.
And we did publish the aging ones. That was also published by Jose Marquez Lopez in Journal Comparative Neurology the year before we published, or 2 years before we published the AOF study.
Gotcha. Okay. We have another question here. Since the ovaries also express angiotensin, what is the effect on your studies?
Well, for our model of hypertension, the angiotensin in the pump is subpressor, and the animals get the same, and it's over a period of 2 weeks. We haven't seen any differences in the levels of angiotensin in either the males, the overectomized females, and the VCD females in terms of the levels of, you know, the systolic blood pressure reading.
Okay.
But we, you know, it's interesting to think about. But
for sure. Another question here is, do any other regions in the brain, sorry, and let me just read this, especially, sir. Are you looking at any other regions in the brain, particularly around the circumventricular organs, in your studies?
So for these studies, they actually started out as part of a program project with Costantino Idacola, Robin Davison, and Jenny Pickle being Costantino's the head of the program. And Robin Davison was actually looking at the subfornical organ. Jenny Pickle was looking at the we had other studies where we looked at nucleus of solitary tract and the rostroventrolatum medulla. In these particular ones, we actually have banked all the tissue from the other brain regions, but we haven't looked at them. We were mainly focusing on the PVN.
Mm-hmm. But it's good that you banked it. You can maybe have a look at it.
Oh yeah, so that's why all these, the nice thing about all these studies that I'm showing you, they were all in the same animals. So it also eliminates the inter-animal variability.
Mm-hmm. Yeah, that's really awesome. Good for longitudinal studies. Okay, we have a follow-up question to the first question. Since the peri and post AOF time points are quite different in terms of age, she's wondering if the results would be different if they age matched by testing day rather than by VCD initiation date. Like, do you think the natural aging would have an effect on the results?
I do. We have not looked at that, but these studies were originally designed to actually separate the aging component from them, so we could actually see what happens with just the hormonal fluctuations. But I do believe adding back age would also have an effect. And if I were to do these again, I would probably even maybe take older animals closer to 10 months to a year, inject them with VCD, and then see how they are affected by at the peri or post AOF time points.
Mm-hmm. Awesome. Another idea for a future study.
Yeah.
There's so much to do. And so
we had that in one grant proposal at one point, and then the reviewers thought it was too much, and we're like, all right, let's just go back and just separate that out.
Yeah. Yeah, that makes sense. Awesome. Okay, we have one last question here for you.
I see two more, actually.
Oh yeah, there's tons. I don't think we'll actually be able to get to all of them, just based on the time.
So okay, all right.
I'm going to jump into this one. You focused on the role of ER beta.
Okay.
Is there any impact of changes in progesterone or pituitary LH and FSH feedback on the hypothalamic circuitry?
We haven't looked specifically at feedback, but it is well known from the VCD literature that there are changes in LH and FSH after you in the AOF model that sort of correspond to what happens in women, and it's in the supplemental part of our first paper with Jose Marquez Lopez, where we looked at some of these other hormones. We did focus on ER beta rather than ER alpha to begin with, because other studies had shown that if you knock out the receptors in different aspects of the circuitry, the autonomic circuitry, that ER beta was the important estrogen receptor at that point.
Okay, fantastic. Terry, I want to thank you so much for your time today and your expertise and sharing your work with us. Really appreciate you and your time, and I hope that you had fun.
Great, thank you. I did. And if anybody can email me if they have more questions.
Absolutely. Thank you again.
Okay, thank you.
And I also wanted to thank our sponsor for today's event, Allzet, for supporting this series and research in the field of women's health. A recording of today's event will be available soon, so keep an eye out for that email. And we'd love to get your feedback in our survey, which will appear at the end of this webinar. You can tell us what topics you'd like to see for the next series. As we conclude the Women's Health series, we'd like to thank our partners at the American Physiological Society and the Society for Women's Health Research for this diverse and engaging program, and also all of our incredible speakers. A huge thank you to them for generously sharing their time and their exceptional work with us. In closing, we hope that you enjoyed this Inside Scientific webinar and series, and we will see you next time.